Welcome to Biology at Priyojit, my dear viewers. Today we will uncover post-translational modifications, the molecular finishing touches that transform simple proteins into powerful functional molecules. Post-translational modifications can be introduced as PTMs, and they are basically chemical changes in protein which occur after translation. This process increases protein diversity and the functionality of proteins. The process also helps in regulation of enzyme activity, gene expression, signal transduction, protein stability and localization. The image showing various kinds of post-translational modifications, that is hydroxylation, ubiquitination, lipidation, schumylation, phosphorylation, acetylation, methylation, glycosylation, and the formation of disulfide bonds within proteins. So, according to our syllabus, now we will start today. Firstly, methylation. Methylation is the process, is a chemical changes that involves the addition of methyl group to a protein. Here, the amino acids are modified are lysine, arginine, and rarely histidine can be methylated. Enzymes involved protein methyl transferases is the enzyme, and the methyl group is received from a methyl donor molecule which is S-adenosyl methionine. So this is the reaction, a methylation reaction showing the methylation of proteins with the help of the enzyme protein methyl transferase, and here the methyl group donor is S-adenosyl methionine or SAM, and this is the methylated protein. There are types of methylation: monomethylation, that is methylation of single methyl group, addition of single methyl group; dimethylation means addition of two methyl group; and trimethylation involves addition of three methyl groups. And the functions of methylation involves it regulates gene expression, it alters protein-protein interaction, it modifies chromatin structure in case of DNA, and it plays a role in epigenetic regulation. Histone 3 methylation, that is H3K4, H3K9, and regulation of transcription factors. There are examples of methylation. Clinical significance: we know that abnormal methylation can cause to induce cancer, transformation of normal cell into cancerous cells, and also it promote neurological disorders. So this is the image showing the arginine methylation, and this is lysine methylation. Acetylation is the process of an addition of acetyl group, which is negatively charged group, to protein. Lysine, that is a which has a epsilon amino group and N-terminal amino group. These amino acids can be acetylated. Histone acetylate acyl transferase or HATs, and histone deacetylases or HDAC can remove the acetyl group. Functions: they neutralizes positive charge of lysine, they weakens DNA histone interaction, they increases gene transcription, and they regulates enzyme activity and protein stability. The examples include histone acetylation, that is euchromatin formation, and acetylation of p53 tumor suppressor protein. So why it has been stated that acetylation process weakens DNA histone interaction? Because we know that DNA is negatively charged and histone is a positively charged protein which contains positively charged amino acids also. So if a negatively charged acetyl group is attached with the positively charged histone protein, then the protein will be converted into negatively charged, and then it will repel the negatively charged DNA molecule. So the DNA histone interaction will be weakened. So now we have to know the biological significance of protein acetylation, that is it regulates gene regulation, cell cycle control, memory, and the learning process. Phosphorylation definition: addition of a phosphate group to a protein, that is PO4 3- group to a protein, is called as phosphorylations. Serine, threonine, and tyrosine amino acids can be phosphorylated with the help of the enzymes known as protein kinase, which can add phosphate. Protein phosphatase remove phosphate, and here phosphate donor is the ATP molecule or GTP molecule can be found. It changes protein conformation. This is the image showing the basic process of protein phosphorylation with the help of the kinase enzyme and utilizing the ATP as a phosphate donor. And phosphatase removes the phosphate group from the phosphoprotein, and then ADP will be converted into ATP. So examples include glycogen phosphorylase activation, MAPK signaling pathways, etc. tyrosine kinase receptors also. Clinical importance promote, that is the dysregulation of phosphorylation process leads to cancer, diabetes and autoimmune disorders. The image showing the phosphorylation event in proteins. Hydroxylation, that is addition of a hydroxyl group, bond OH group to amino acid, is hydroxylation. Here proline, lysine amino acids are involved in the hydroxylation process. Prolyl hydroxylates and lysyl hydroxylase is involved in proline and lysine phosphorylation, hydroxylation respectively. Requirements, they need vitamin C, ascorbic acid as their cofactor or coenzyme. If Fe2 plus oxygen is also required for the enzymatic activity. This hydroxylation process also stabilizes protein structure. They are essential for collagen maturation. They helps in triple helix formation within DNA. So examples, hydroxyproline is found in collagen and hydroxylysine is found in connective tissue. Clinical importance, vitamin C deficiency causes scurvy, and that occurs due to weak connective tissue and poor wound healing. So this is the image showing the hydroxylation of proline and lysine with the enzyme hydroxylase, and they respectively converted into hydroxyproline and hydroxylysine. Glycosylation, that is attachment of the carbohydrate or glycan chains to protein, is called as glycosylation. There are two types of glycosylation: N-linked glycosylation, where attachment to asparagine, that is the amino group of the asparagine when glycosyl moiety is attached. It is found in endoplasmic reticulum. The consensus sequence is the asparagine, that is X amino acid, any kind of amino acid, then serine or threonine, and this can be glycosylated. Asparagine residue can be glycosylated. O-linked glycosylation, that is attachment to serine and threonine, where bond hydroxyl group is present, and that oxygen atom of the hydroxyl group can be glycosylated. It occurs in Golgi apparatus. So what we found, N-linked glycosylation occurs in endoplasmic reticulum and O-linked glycosylation occurs in Golgi apparatus. So what is the functions of glycosylation? It involves, it help promote proper protein folding, increase protein stability, it promotes cell recognition, influence immune response, also regulate protein targeting and trafficking. So examples include immunoglobulins, hormones such as erythropoietin, cell surface receptors. Those are examples where glycosylation can be found. In biological importance, blood group antigens, cell adhesion molecules and defects cause congenital disorders of glycosylation. So in conclusion, post-translational modifications that we have studied today are essential molecular refinements that fine-tune protein function and protein activity. These modifications ensure precise cellular regulation and effective biological responses. So my dear students, keep exploring the dynamic world of molecular biology. Thank you.